This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy. We go deep and lightly surface the big ideas.
If a surgeon were to open up your skull right now, they wouldn't find a color-coded map.
Right, no labels in there.
Yeah, there isn't a little region of gray matter with a brightly lit, microscopic sign that says language department.
Which is unfortunate for neurosurgeons, honestly.
Seriously. Because for decades, we've treated the human body like a predictable piece of engineering.
Right.
I mean, you break your arm, the X-ray shows a jagged white line, and the doctor points to a universal schematic.
Blueprints, yeah.
Exactly. The heart is here, the lungs are there, the femur connects to the pelvis. But when you step into the world of neurobiology, and specifically the neurobiology of language, that universal schematic just kind of disintegrates.
It completely falls apart.
Because the physical geography of where your words, your grammar, and your understanding live is, it's as uniquely yours as your fingerprint.
It really is.
So if your language network is completely different in shape and location from the person sitting next to you, how do scientists actually find it?
Well, historically, finding that unique linguistic fingerprint has been a really frustrating exercise in approximation.
Not like guessing, basically.
Pretty much. I mean, the field has spent over a century working with these blurry, averaged out maps that just wash away all those individual differences.
Right.
But a recent breakthrough has completely upended how we approach this. It turns out we don't need to force a universal map onto your unique brain. We just need to know how to listen to it.
And that brings us to the focus of today's deep dive. We are looking at a massive 2026 neuroscience paper. This was published in Nature Communications by researchers Cory Shain and Evelina Fedorenko.
It's a huge paper.
It's giant. It's titled Language Network in the Individualized Functional Connectomes of 1,199 Human Brains Doing Arbitrary Tasks.
Rolls right off the tongue.
Doesn't it just? But our mission today is to explore this profound paradigm shift. We're going to look at how these scientists figured out a way to map the exact, highly individualized language center of your brain without ever asking you to speak.
Or read. Or listen to a single word.
Right. They managed to map a function by ignoring the function entirely.
Yeah.
They just eavesdropped on the background hum of your brain while you do something totally unrelated like, I don't know, solving a math problem.
Or just staring blankly at a screen.
Okay, let's unpack this, because to appreciate the magnitude of this, we kind of have to look at how stubborn the old methods were.
Oh, incredibly entrenched. What's fascinating here is that this paper doesn't just introduce a new technique, It actively challenges 150 years of deeply held assumptions about how we localize human cognition.
We've been trying to force this highly dynamic network into a rigid box.
A very rigid macroanatomical box.
So let's trace the origins of that box. Where did this stubborn idea that language lives in this exact 1-inch square of the brain for every single human being actually come from?
Well, it starts back in the 1860s with a French physician named Paul Broca.
Right, Broca's area. We all learned about that in Psych 101.
Exactly. And the prevailing method at the time was lesion-based symptom mapping. Broca had a famous patient, Louis-Victor Leborgne, who actually went by the nickname Tan.
Why Tan?
Because Tan was the only syllable he could articulate. He had severe expressive aphasia.
Oh, wow.
Yeah. So when Tan died, Broca autopsied his brain and found a significant lesion, basically tissue damage from neurosyphilis, in a very specific spot. The posterior inferior frontal gyrus of the left hemisphere.
So Broca sees a hole in that specific spot, and the guy couldn't speak, so he connects the dots.
Right. Broca presented this, and the scientific community just extrapolated this massive conclusion. They said, that specific physical location is the absolute center of speech production.
And then Wernicke comes along later.
Yeah, a few years later, Carl Wernicke found lesions further back in the temporal lobe that were associated with language comprehension deficits.
So based on just a handful of brain-damaged patients in the 19th century, we drew a hard boundary on a map and said, this is Broca's area, this is Wernicke's area, and we just expected everyone's brain to perfectly follow that cartography.
We absolutely did. It created a textbook model that, frankly, we still teach today.
It's a nice, neat story.
It is. It's a very attractive idea because it suggests the brain is modular, like the motherboard of a computer.
Plug and play.
Exactly. But as imaging technology advanced, especially with fMRI in the 90s, the cracks in that modular theory started to really show. The researchers in this new paper argue that searching for a universal language area tied to gross anatomy is what they call an ill-posed question.
Okay, when scientists call a 150-year-old pursuit an ill-posed question, that's usually academic code for we've been banging our heads against the wrong wall.
That is exactly what it means.
So what makes the question inherently flawed?
It comes down to a biological reality called weak tethering.
Weak tethering.
Yeah. So if you look at the surface of the human brain, it's covered in all these folds and ridges, the sulci and the gyri. These are our macroanatomical landmarks.
The physical hills and valleys of the brain.
Right. And for a long time, the assumption was that functional areas were strictly tethered to those physical landmarks. Like if a ridge is in the same general spot on my brain as it is on yours, it must do the exact same job.
But that's not true.
It is simply not true for higher order cognitive networks like language. The language network is only loosely or weakly tethered to those folds. In your brain, the language network might spill over a specific sulcus, but in my brain, it might stop 2 centimeters short of it.
Wait, if the network isn't anchored to the physical landscape, how did standard fMRI studies ever claim to find a universal language center? I mean, I've seen those colorful brain scan images in articles where a specific blob is lit up, claiming to show, like, your brain on language.
Oh, the famous lit-up blobs?
Yeah. How are they generating those blobs if everyone's brain is physically different?
They generate them through a mathematical process called normalization. And this inevitably leads to group averaging.
Group averaging. Okay, how does that work?
Well, because every brain is a different size and shape, early neuroimagers had to figure out a way to compare them. So they created these standard spatial templates. The most common one is the Montreal Neurological Institute, or MNI space.
Okay.
When they take a scan of your brain, the software literally stretches, squishes, and warps your unique brain volume to fit into this generic universal template.
That sounds aggressive.
It is. And once they've warped 100 different brains to fit the exact same mold, they average the functional activity altogether.
Wait, so standard brain mapping is like trying to figure out the layout of a typical house by banding together the blueprints of a mansion, a studio apartment, and an RV.
That is a phenomenal analogy.
I mean, no wonder the average map is a mess. If you do that, the resulting map is going to put the kitchen light switch hovering in the middle of my bathtub.
Exactly. That is the perfect mechanistic way to look at it. When you average those 100 brains together in MNI space, the sharp, highly distinct boundaries of the individual's language network are completely washed out.
They just get so mirrored.
Yes. The peaks of activity are blunted, and the borders smear out into all the neighboring networks. You end up with a giant, blurry blob that doesn't actually represent the true functional anatomy of any single human being in that group.
It's an illusion.
A mathematical illusion. The source paper emphasizes that by focusing on the population level, these standard analytic approaches completely miss the reality of the individual.
So anatomical variability, the fact that my brain folds differently and my networks are shifted, that's the first major hurdle. But the paper points out a second, arguably more frustrating barrier.
The conceptual disagreement.
Right. Before we even get to the fMRI scanner, the people designing the experiments are fighting a theoretical war over what they are actually trying to measure.
Oh, the field of linguistics is highly fractured. For decades, researchers have been locked in these bitter debates over the construct validity of language.
Like what language actually is.
Exactly. What is the irreducible core of human language? Is it syntax?
Yeah.
You know, the abstract hierarchical rules of grammar as championed by theorists like Noam Chomsky?
Right.
Or is language fundamentally about semantics and usage, like how we extract meaning from context? When you move from theoretical linguistics into an fMRI lab, you have to operationalize those theories.
But to build a test.
Right. You have to design a task to make the language network light up.
Okay, give me an example of how you operationalize language in a scanner. Like, how do you force a brain to do language while someone is lying perfectly still in a magnetic tube?
The historical gold standard, which Fedorenko's own lab actually pioneered years ago, is the functional localizer task.
Okay.
The most common version is a reading task. You show a participant a series of complete, meaningful sentences, like, "The weary traveler finally reached the inn."
Makes sense.
Then, as a control, you show them lists of pronounceable non-words. Gibberish that follows English phonotactic rules but means nothing, like blicket, wugged, the fep.
Bliget, wug, the fep. It sounds like Lewis Carroll's Jabberwocky.
It's very Jabberwocky.
So the logic is, Reading a real sentence uses vocabulary and grammar.
Uh,
Reading the gibberish requires you to sound out the letters, so it uses basic visual and auditory processing, but it doesn't trigger the higher level meaning or sentence structure.
Precisely. You subtract the brain activity of the gibberish from the brain activity of the real sentence, and whatever is left over the mathematical difference is your language network.
That feels pretty smart, actually. But let me guess, the theorists hated it.
Theoretical linguists despise this approach.
Why?
They argue that a task contrasting full sentences with gibberish is incredibly broad. It captures syntax, semantics, morphosyntax, lexical access, everything all at once.
It's too buddy.
Right. The critics argue that this task fails to isolate the purely structural, grammatical computations that they believe are the true essence of language.
Wait, isn't that just a turf war over definitions?
Yeah.
I mean, if a researcher believes language is purely about grammar, they'll design an experiment that hyper-focuses on grammar, find a specific region that lights up, and declare they've found the true language center.
Yep.
Then another researcher who cares about vocabulary will design a completely different test, find a different region, and claim they are right. The theoretical bias just dictates the physical result.
You've just diagnosed the exact crisis in the field. When your diagnostic tool requires the patient to perform a highly specific cognitive task, the assumptions baked into that task dictate the map you produce.
That is a methodological nightmare.
It is. As the source paper explicitly states, it is nearly impossible to pursue a cumulative, objective neurobiology of language when there are core disagreements about how language should even be conceptualized or tested.
So to map the brain, we need to know where things are, but everyone's map is physically different.
Yeah.
And to figure out where things are, we need to test them. But nobody agrees on what the test should be.
It's quite the paradox.
We're going to take a quick break here, but when we come back, we're going to dive into how this new massive study managed to completely bypass this paradox. Stick around. PRX station break. Welcome back to the deep dive. So before the break, we laid out this massive problem. Standard brain mapping is blurry, and task-based tests are completely biased. How do you escape that loop?
You escape it by abandoning the task entirely.
Just throw the test out.
Exactly. If the task is the source of the bias, you have to find a way to map the network without asking the brain to do anything linguistic. And that leads us to the methodological breakthrough of this paper. Individualized functional connectomics, or IFC.
Connectomics, the study of how the brain connects to itself.
Yes.
So instead of looking for which part of the brain is like working hard during a reading test, IFC looks at the baseline communication between brain regions over time.
That's it exactly. To understand how IFC achieves this, we need to look at the actual physics and biology of what an fMRI machine is measuring.
Okay, let's break down the mechanics, because this is the absolute crux of how they are eavesdropping on the brain.
Right. An fMRI doesn't measure neural electricity directly. It measures the BOLD signal.
Bold, like D-O-L-D.
Yes. The blood oxygen level dependent signal.
Okay, so when a neuron fires in my temporal lobe, what is physically happening with the blood, and how does a giant magnet detect it?
So neurons don't have their own internal reserves of energy. When a population of neurons fires to process a thought, they rapidly consume local oxygen and glucose.
They burn through their fuel.
Exactly. To replenish this, the brain initiates a process called neurovascular coupling. Astrocytes, which are these star-shaped glial cells that act as intermediaries between neurons and blood vessels, they detect the neural activity and signal the local capillaries to dilate.
So the pipes open wider.
Yes. But the circulatory system actually overcompensates. It sends a massive rush of highly oxygenated blood to that specific area, way more than the neurons actually just consumed.
Okay, so you get a localized flood of oxygen-rich blood. Why does the MRI machine care about oxygen?
Because of the magnetic properties of hemoglobin.
Oh, right. Iron in the blood.
Exactly. Hemoglobin carrying oxygen is diamagnetic. It doesn't really interfere with the fMRI's magnetic field, but deoxygenated hemoglobin is paramagnetic. It actively disrupts the magnetic field, which causes the signal to drop.
Oh, I see.
So because the brain oversupplies oxygenated blood to the active area, the ratio of oxygenated to deoxygenated hemoglobin suddenly spikes. That relative decrease in magnetic disruption is what the fMRI scanner picks up as a localized signal increase.
And that is the BOLD signal.
That is the BOLD signal.
Okay, so the fMRI is basically just a highly sensitive blood flow tracker. But here is the critical leap. In traditional experiment, you give the subject a task to intentionally trigger that blood flow in a specific area. But you said IFC doesn't use a task.
Right, no task.
It looks at the spontaneous, low-frequency fluctuations of that blood flow while the brain is supposedly at rest. But why is the blood fluctuating if I'm not doing anything?
Because the brain is never truly at rest. Even when you aren't actively speaking or reading, your neural networks are maintaining a baseline level of coordinated activity.
They're just humming.
They are humming. If we think of brain regions as, say, a highly coordinated symphony orchestra, even when they aren't actively playing a piece of music for the audience, which would be the task, the musicians are still quietly tuning their instruments.
They're breathing in sync, making micro-adjustments.
Exactly. Regions that serve the same overarching function, like language, exhibit synchronized fluctuations in their blood oxygen levels over time. Their bold signals rise and fall together in this slow, correlated dance.
Here's where it gets really interesting to me. It's like figuring out who the HR department is in a massive corporation just by watching who goes to lunch together.
Yes.
Like, you don't need to wait for them to actually fire someone to know they're a cohesive unit. You just watch who gossips by the water cooler.
That is a perfect analogy. IFC is mapping those intrinsic social cliques of the brain. Now, earlier IFC studies did attempt to map the language network, but they were severely limited.
Oh, so?
Well, they usually involved fewer than 20 subjects, and they relied exclusively on resting state scans, where subjects were explicitly told to lie still, keep their eyes open, and try not to think about anything in particular.
Which is an impossible instruction. If you tell me to think about nothing, my internal monologue immediately fires up. I'm like, am I thinking about nothing? What's for dinner? My nose itches. How much longer am I in this tube?
Right.
My language network is absolutely running during a resting state scan.
Exactly. And relying on dedicated resting state scans still requires expensive dedicated scanner time. So the radical hypothesis that Shain and Fedorenko tested in this 2026 paper was this. The synchronized hum of the language network is so robust, so intrinsically wired together, that it should be detectable not just during rest, but during any cognitive state.
Any state.
They hypothesized that IFC is entirely task agnostic.
Meaning they should be able to find the language network's synchronized hum, even if the person is actively doing a completely different, highly demanding task like a math problem.
Yes.
So the math network might be firing loudly to solve the equation, but underneath that, the language network is still whispering to itself in its own specific frequency.
Precisely. And to prove this, they didn't just run a small trial. They leveraged the task-agnostic nature of IFC to conduct a study at an unprecedented scale. They went straight into the archives.
Because they didn't need new tests.
Exactly. They extracted every single usable fMRI session collected in the Fedorenko lab between 2007 and 2024.
17 years of archival fMRI data. Because they didn't need the subjects to be doing a specific language task, they could pull data from memory experiments, visual perception experiments, executive function experiments. It just didn't matter what the original researchers were studying.
It didn't matter at all. They compiled 1,957 fMRI sessions from 11,199 unique human brains.
That is a staggering amount of data.
It represents thousands of hours of imaging data. It is 2 orders of magnitude larger than any prior IFC study of language. But having a mountain of data is only the first step.
Right, because now you have to sort it.
The monumental challenge is the mathematical analysis. How do you find the specific resonance of the language network in a sea of overwhelming biological noise across 1,000 different brains?
And this is where we need to dive into the algorithm. The paper details a fully automated machine learning process to sort this out. They use a technique called K-means clustering.
Yes, K-means.
Let's walk through the exact mechanics of how a computer looks at an fMRI scan and automatically finds the networks, because the scale of this math is kind of mind-blowing.
First, we have to understand the data structure. A single fMRI scan breaks the brain down into tiny 3-dimensional cubes called voxels.
Like 3D pixels.
Exactly. In this study, they analyzed roughly 134,000 voxels across the gray matter of each individual brain.
Okay, 134,000 tiny cubes.
And for every single one of those 134,000 voxels, the scanner records a time series vector. It measures the Boldy signal intensity every couple of seconds over a period of several minutes.
So if the scan is 5 minutes long, taking a picture every 2 seconds, each of those 134,000 voxels has a unique string of 150 numbers attached to it, just representing how its blood flow went up and down over time.
Yes. So the k-means algorithm takes all 134,000 of those time series vectors and plots them in a highly complex, multidimensional mathematical space.
Okay.
Its goal is to find groups or clusters of voxels whose time series vectors look highly similar. It calculates the Euclidean distance between these vectors.
So voxels that act similar are grouped closer together.
Right. Voxels whose blood flow rises and falls in tight synchronization are mathematically closer together in the space. The algorithm groups them into k number of distinct clusters.
But the paper notes a significant vulnerability with standard k-means clustering. It can suffer from the local optimum problem.
It definitely can.
If I understand this correctly, co-means has to start somewhere. It throws down a few random anchor points called centroids and starts grouping the voxels around them. But if those initial anchor points are placed in weird spots by pure chance, the algorithm will confidently draw boundaries that are completely wrong, and it won't even realize it made a mistake.
That is the exact mathematical vulnerability. It finds a solution that looks good locally, but misses the optimal global solution. To solve this, the researchers used a brute force statistical approach.
Brute force is my favorite kind of math.
It's very effective. They used 100 random initializations. For every single brain session, they didn't just run the algorithm once. They randomized the starting anchor points and reran the entire clustering process 100 separate times.
It's like trying to find the natural social cliques at a massive high school. You randomly assign seating charts in the cafeteria 100 days in a row, and you just observe who consistently abandons their assigned seat to go sit with the exact same group of friends.
That's a great way to visualize it.
Right. If voxel A and voxel B end up in the same cluster 99 times out of 100, regardless of how the algorithm initially sorted them, you have immense statistical confidence that those two pieces of brain tissue are intrinsically functionally connected.
Exactly. That repeated permutation creates a highly robust probabilistic map of the networks. But once the algorithm has successfully clustered these 134,000 voxels into, say, several dozen distinct synchronized networks, there is still one major hurdle.
The computer doesn't speak English.
Nope.
The algorithm has grouped the voxels based purely on their mathematical correlation. Doesn't know what those networks actually do, it just knows they hum at the same frequency. So how do they slap a label on these anonymous clusters and confidently say, this one is the language network?
They use a spatial matching step against reference atlases.
Okay, atlases.
Over the decades, population-level imaging has given us rough, probabilistic maps of where major networks generally tend to sit. The researchers used 16 of these reference atlases, including the General Language Atlas, the Default Mode Network, the Frontoparietal Network, and the Auditory Network.
So they use the old blurry maps just as a rough guide to name the sharp new clusters.
Exactly. The algorithm takes the highly distinct, individualized clusters it just found in your specific brain and calculates the spatial overlap with those generic atlases. If one of your individualized clusters physically overlaps heavily with the generic language atlas, the algorithm assigns it the label LangFC.
So they let the raw, task-free data organize itself naturally, based purely on synchronized blood flow, and then they play a quick game of spatial matching to identify which cluster is which.
That's the process.
And when they finally extracted this newly labeled individualized LangFC network, what did it actually look like? Did it solve the blurring problem we talked about in Act 1?
Oh, it obliterated the blurring problem.
What?
The LangFC network they discovered is astonishingly distinct from abutting networks. This is a critical finding that validates the entire methodology. If you look at the temporal lobe, the language network is situated right next door to the auditory network.
Which makes sense. Hearing and language.
Right. It is also adjacent to a subcomponent of the default mode network, known as DNB, which handles theory of mind, our ability to model the social intentions and thoughts of other people.
Right, so physically, the understanding grammar department is sharing a wall with the hearing noises department and the social empathy department.
They're very tight neighbors.
And in the old group average maps, you said those three departments essentially smeared into one giant functional blob.
They did. But when derived through individualized functional connectomics, the boundaries between them snap into sharp focus. The paper demonstrates that the temporal correlation between your individualized language network and your auditory network is near zero, sometimes even negative.
Wait, really? Their blood flow fluctuations do not synchronize at all?
Not at all.
Which logically has to be true. I mean, if I'm walking down the street and I hear a fire engine siren, my auditory network needs to process that immediately. But a siren has no grammar. It has no vocabulary. My language network shouldn't care about it.
Exactly. And the same boundary sharpness exists with the theory of mind network. The separation of these networks explains decades of false positives in neurobiology.
How so?
For years, scientists saw activity in the temporoparietal junction, the TPJ, during language tasks and assumed the TPJ was part of the language network.
Why did it light up during a language task if it's not part of the language network?
Because of what the subjects were reading. If you put someone in a scanner and have them read a complex story about two characters arguing, you are absolutely engaging their language network to parse the vocabulary and syntax.
Right.
But you are simultaneously engaging their theory of mind network as they try to understand the character's motivations and emotions. The two networks are working simultaneously on different aspects of the stimulus.
And because group averaging blurred the physical boundaries, researchers just assumed it was all one big linguistic response.
Exactly. IFC proves that the language network systematically excludes those temporoparietal areas. They belong to social cognition.
That is a staggering cleanup of the scientific literature.
It is massive.
But wait, if the LangFC network was defined entirely by its background hum during arbitrary tasks, how do we know it actually handles language? Maybe it's just an artifact of blood flow. Did they actually test it?
They did, and the results are definitive. They evaluated this newly mapped LangFC network against actual task-based fMRI data.
Okay.
They found the network in an individual while that person was doing a non-linguistic task, like a spatial working memory puzzle. Then, in a separate session, they gave that same person a traditional language localizer task reading sentences versus gibberish.
And what happened?
The network that lit up during the reading task mapped perfectly onto the LangFC network they had found during the memory puzzle.
That feels like a massive contradiction. How can a network be defined by doing nothing related to language, yet perfectly predict what happens when the brain does everything related to language?
Because the underlying functional architecture doesn't disappear when you aren't using it. The resonance is intrinsic to the anatomy.
It's always there.
It is. The paper literally ran a sub-analysis where they completely excluded any scanning session that contained any language stimuli whatsoever. No reading, no listening to speech. They only fed the algorithm data from math tasks, face perception tasks, and rest.
And it still found it.
The LangFC network still emerged from the data, beautifully intact and structurally identical to the network found during reading tasks. The language network is a fundamental, permanent feature of your individual connectome, constantly humming its own unique tune.
Okay, this leads us to perhaps the most controversial finding in the paper. They call it the indivisibility of the network. And this is where I think they're picking a direct fight with the textbook models of linguistics we discussed earlier.
Oh, they are absolutely picking a fight, and they have the data to back it up. This raises an important question about how we fundamentally view language. Let's return to the classic Psychology 101 model.
The Broca and Wernicke assembly line.
Exactly. The traditional view is highly fractionated. It posits that the brain operates like an assembly line. Syntax, the structural rules, is handled by a specific module historically associated with Broca's area in the frontal lobe. Semantics, the meaning of individual words, is handled by a separate module, often associated with Wernicke's area in the temporal lobe.
Right. One worker on the assembly line identifies the nouns, passes the conveyor belt to the next worker who conjugates the verbs, and passes it to the next worker who checks the hierarchical sentence structure. It assumes the network is made of specialized subcomponents.
Right. So to test if this assembly line model is true, the researchers pushed their k-means clustering algorithm to its absolute limits. Remember, k is the number of clusters the algorithm creates.
Yeah.
They ran the analysis setting k to 10, then 50, then 100, and all the way up to 200 distinct networks.
They gave the algorithm 200 opportunities to slice and dice the brain into smaller and smaller subdepartments.
Exactly. If the textbook model is correct, if syntax and semantics are truly distinct functional modules, then as you increase the granularity of the clustering, the overarching language network should fracture. It should split.
Because they're doing different jobs.
Right. The voxels in the frontal lobe representing syntax should form their own cluster, and the voxels in the temporal lobe representing semantics should form a separate cluster.
Let me push back on this methodology for a second. Is fMRI even capable of seeing a fracture like that? I mean, the Boldy signal is measuring blood flow, which is inherently slow and sluggish compared to the millisecond speed of actual neural electricity. Are we sure the language network isn't actually fractured, but the fMRI is just too blurry to see the dividing line?
That is the exact critique a skeptical neuroscientist would raise, but the researchers anticipated it. We know fMRI has the resolution to see fractures because we see it happen in other networks.
Like which ones?
When they turn the dial up to 200 parcellations, the motor network fractures beautifully into distinct subnetworks for the hands, the feet, and the face.
Oh, yeah.
The visual network fractures into distinct subregions for central versus peripheral vision. The spatial resolution of fMRI is absolutely sufficient to detect these functional subdivisions.
But the language network didn't fracture.
It flatly refused.
Yeah.
Even when forced to chop the brain into 200 tiny networks, the language network remained a single, unified cluster.
Wow.
The baseline bold defluctuations in the frontal lobe and the temporal lobe were so tightly correlated, their background hum was so perfectly, inextricably synchronized, that the algorithm kept them grouped as one cohesive unit.
So what does this all mean for the classic Broca's area and Wernicke's area we all learned about in Psychology 101? Are you telling me that slicing language up into grammar regions and vocabulary regions is just a myth we imposed on the brain rather than how the brain actually works?
To quote the authors directly, this result reduces the plausibility of any claims about sharp network internal functional subdivisions at scales detectable using fMRI.
So it's a myth.
It strongly implies that the entire network operates as a single, tightly integrated computational space. It isn't an assembly line, it's a holistic system. Every part of the network cares about syntax, and every part cares about semantics. They process the linguistic input together simultaneously.
It implies that we've just been projecting our own human obsession with categorization onto biology. We invented the concepts of nouns, verbs, and syntax to teach language in school, and then we desperately went looking for those exact categories in the brain tissue.
We wanted the brain to look like a textbook.
But the brain doesn't care about our grammar textbooks. It just processes language as a unified phenomenon.
It is a profound philosophical shift. The term language network ceases to be just a label we apply to whatever lights up during a reading test. It becomes a purely descriptive term for a naturally occurring, independent anatomical structure that exists intrinsically within you, humming away whether we test it or not.
That is deeply profound. We're going to take one more quick break. When we return, we are going to get incredibly practical. What are the real-world superpowers this new map gives us? Stick around. PRX station break. All right, we are back. I am thoroughly convinced by the theoretical elegance of this, but mapping the indivisible, individualized network purely from background noise. What is the practical utility of this?
It's massive.
If my language network is completely unique to me, doesn't that make standardizing medical treatments or research incredibly difficult? I want to explore the superpowers this gives us in the real world.
The practical utility is actually staggering, and the first major superpower comes down to the sheer data efficiency of this new method. In standard practice, identifying a patient's language network with a functional localizer task takes about 10 to 15 minutes of exhausting, active cognitive effort inside the scanner.
Which, as we discussed, is expensive and highly demanding on the subject.
Very demanding. The researchers wanted to know the absolute minimum amount of scanning time required to accurately pull out this individualized LangFC map using just background noise.
So they ran a stress test.
Exactly. They took a subset of 53 participants who had been scanned extensively over 5 hours of data each. They used that massive amount of data to create a gold standard map for each person. Then, they systematically fed the algorithm less and less data to see when the map would break down.
How low could they go?
They found that while stability is optimal around 5 runs of scanning, the individualized language network can be accurately mapped from just a single run of fMRI data.
A single run. In standard fMRI protocols, how much time is a single run?
Between 3 to 5 minutes.
3 minutes. You put someone in an MRI tube for 3 minutes. Tell them just to lie there, and the algorithm can extract a highly precise, biologically accurate map of their unique language center with sharp boundaries that isolate it from auditory and social cognition.
Precisely. And because it requires no task, because it is entirely task agnostic, it unlocks the second superpower.
Which is?
Retrospective analysis. This is the archaeological aspect of the breakthrough.
Right. If you don't need a task, you can use old data.
Exactly. There are open-source neuroimaging databases around the world containing hundreds of thousands of hours of fMRI data. Historically, if a researcher wanted to study the neurobiology of language, but a specific data set didn't include a reading or listening task, that data was totally useless to them.
Because they had no way to know where the language network was located in those specific subjects.
Right. But now they have the skeleton key. They can take an fMRI dataset from a 2015 study on visual memory, run this algorithm on the raw data, and retroactively map the exact individualized language networks of all those participants.
It democratizes neuroscience. Researchers no longer need massive grants to book hundreds of hours of scanner time just to find the language network. They can extract the network from massive pre-existing open-access data sets. It accelerates the pace of discovery exponentially.
It really does. It opens up so many doors.
Let's pivot to the clinical applications, because this feels like it has immediate life or death utility. In neurosurgery, the physical map of the brain is the only thing standing between a successful operation and a catastrophic outcome.
Absolutely. If a patient requires a craniotomy to remove a glioma, a brain tumor in the frontal or temporal lobes, the neurosurgeon's highest priority, secondary to removing the pathology, is avoiding what they call eloquent cortex.
Eloquent cortex.
Yes, they must not damage the tissue that controls movement, vision, or language. If the surgeon cuts into your language network, you could wake up with permanent aphasia. You might lose the ability to speak or to understand your family.
And as we established in Act 1, the surgeon cannot rely on a textbook map because the patient's language network is weakly tethered. It might be 2 centimeters higher or lower than the generic model predicts. It might be distorted by the tumor itself, pushing the tissue around.
Currently, surgeons map the eloquent cortex using preoperative task-based fMRI and then intraoperative awake brain mapping.
Awake brain map.
Yeah, where they literally stimulate the exposed brain with an electrode while the patient is awake and talking on the operating table.
That sounds terrifying.
It is. The preoperative fMRI requires the patient who is likely terrified, potentially cognitively impaired by the tumor, and fatigued to perform complex reading tasks inside the scanner. If the patient is a young child or someone with severe cognitive deficits, those tasks are often impossible to complete reliably.
But with IFC.
With IFC, you eliminate the cognitive burden entirely. You place the patient in the scanner for a few minutes. They don't have to read sentences. They don't have to speak. They can close their eyes and rest.
And the math does the work.
The K-means algorithm eavesdrops on the synchronized blood flow and extract the highly precise, custom-tailored topographical map of their unique LangFC network. It hands the neurosurgeon a personalized GPS guide to that specific patient's brain, minimizing surgical risk and saving crucial diagnostic time.
That is an immediate, massive leap forward for patient care.
Mm.
What about beyond surgery? Are there implications for neural engineering?
Oh, this is where we look toward the frontier of brain-computer interfaces, or BCIs. We are entering an era where paralyzed individuals, people with ALS or brainstem strokes, who are completely locked in, can communicate through electrocorticography grids implanted directly on the surface of their brain.
The devices that literally decode intended speech from thought.
Yes. But the efficacy of those implants depends entirely on spatial precision. You have to place the recording electrodes directly over the neurons that process language formulation. If the language network is highly variable across individuals, knowing exactly where to place that grid is a monumental challenge. If you are off by a centimeter, you lose the signal.
I see where this is going. If we can map their individual holistic language network perfectly, using just a few minutes of background Abuldi signal fluctuations, we generate the exact coordinates for the BCI implantation.
We provide a perfect targeting system for the next generation of neural prostheses. The IFC map tells the engineers exactly where the language nodes are located for that specific patient, allowing for custom-built, perfectly positioned sensor arrays.
Okay, let me try to synthesize this entire journey. For a century and a half, science was trapped by a rigid theoretical map of the brain. We assumed language lived in universally identical zip codes, and when the data didn't perfectly align, we averaged everyone together, creating blurry, inaccurate models that washed out individual humanity.
A very messy history.
And we argued endlessly about whether language was just syntax or semantics, and we designed biased tests that forced the brain to answer questions it wasn't actually asking.
And the solution to all of that historical baggage was to simply stop forcing the brain to perform and start listening to its intrinsic architecture.
By looking at the raw, staggering scale of 11,199 human brains, Shain and Fedorenko proved that by analyzing the low-frequency synchronized fluctuations of blood oxygen levels, the baseline hum of the brain, we can map a highly unique, deeply personal language network in every single individual.
Perfectly said.
We discovered that this network doesn't operate as a fractionated assembly line of grammar and vocabulary, but as a single, tightly integrated, indivisible unit, a computational ecosystem that is distinctly yours.
And that single discovery retroactively unlocks decades of historical fMRI data, drastically reduces the burden on neurosurgical patients, and maps the exact coordinates needed for the future of brain-computer communication. It represents a total triumph of data-driven biological reality over abstract theoretical dogma.
Which leaves me with a final, slightly provocative thought for you to ponder. If your language network is this distinctly yours, operating as one tightly integrated, indivisible island that differs in shape, size, and location from the person sitting next to you, what does that mean for the far future of how we interact with technology?
It's a great question.
If we extrapolate this to the ultimate end game of brain-computer interfaces, think about the classic sci-fi trope from The Matrix. When they want to learn something instantly, they just plug a cable into their skull and download the knowledge. I know Kung Fu or, download the French language module.
Right, the instant upload.
But based on this paper, a universal download cable is biologically impossible. You couldn't just plug a universal French USB drive into my brain because of the geographic location and the internal network topography of where my brain would process that language is utterly different from yours. The software literally wouldn't know where to install itself.
It would just be a mismatch.
Exactly. So will the artificial intelligence of the future have to spend a few minutes eavesdropping on your unique biological frequency first? Will it have to map your specific connectome, calculating the exact spatial coordinates and resonant hum of your individual network, just to find the front door to your mind?
It certainly seems that way.
It seems that the deeper we look into the universal mechanics of human biology, the more we discover just how radically, fundamentally unique each and every one of us actually is. Thank you for joining us on this deep dive into the individualized geography of the mind. As always, this exploration was custom-tailored just for you. Keep listening to the background hum. Heliox is produced by Michelle Brucker and Scott Bleakley. It features reviews of emerging research and ideas from leading thinkers, curated under their creative direction with AI assistance for voice, imagery, and composition. Systemic voices and illustrative images of people are representative tools, not depictions of specific individuals. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other episodes, responding to the content, and checking out our related articles at helioxpodcast.substack.com.